So I'm going to introduce our first speaker. Our first speaker is Dan Ban. So Dan is the Sir Henry Wellcome Postdoctoral Fellow at the Wellcome Center for Human Neuroimaging at the University College London. He completed his PhD at the University of Oxford under the supervision of Professor Christopher Summerfield, working on perceptual and social decision-making in humans. He then did a postdoc with Dr. Stephen Fleming at the Wellcome Center for Human Neuroimaging, examining the neural basis of physician confidence. He's currently studying the neurobiological foundations of flexible social behavior in humans, using behavioral testing, computational modeling, and novel methods for measuring neurochemistry and neurocircuitry in human brain. So I'll turn over to Dan. And
should I start now, or because I think it's scheduled to start 15:15. So you're muted, Ben.
You're right. The introduction was a bit faster than I thought. Just wait a few minutes so you can start us at 9:15 Eastern time, so that everyone who wants to join at the time can actually do it.
Okay. And then, yeah, and I guess you're seeing my shared screen now?
Yes, yeah, we'll see it.
Brilliant. Cool.
Then, in the afternoon, if we want to listen to the talks, we would have to go into the guest room. Is that so?
No, you can just listen normally to the talk. Just make sure that you join as an audience, not as a presenter when you join the stream.
Yes, okay. Thank you.
Okay, so I guess I can get started now. I just want to make sure. Can you see my screen now?
Yes.
Yes.
Okay, brilliant. Yeah. So first of all, thanks to the organizers for putting together this virtual symposium. It's amazing we can take part in this. I'm in the UK, locked up in my house, but it's great that I have this opportunity. So today I'll tell you about our recent work on fast neuromodulation in the conscious human brain. And it's only recently that it's become possible to study fast neuromodulation in humans, so I'll spend a bit of time explaining how we actually did it. I want to start by thanking these two amazing scientists. Ken and Reed have pioneered the study of fast neuromodulation in humans, and it's been amazing to get to work with them and use their methods to study the kinds of cognitive questions that I'm interested in. And I should mention that Ken is currently hiring postdocs, so if you're excited about the work that I'll present today, do get in touch with him. So, as probably most people at this symposium know, dopamine and serotonin are two of the brain's major neuromodulatory systems, and they're crucial for healthy function. Each system originates in distinct nuclei in the midbrain, and they broadcast chemical messages widely throughout the brain through dense projections to both subcortical and cortical structures. Our current understanding is that the systems support basic physiological processes both at the neuronal and at the circuit level. We also believe that the systems participate in fundamental cognitive processes from motivation to decision-making and learning. And consistent with these broad roles, both systems have been linked to a wide range of clinical conditions. For example, Parkinson's disease, associated with the loss of midbrain dopamine neurons, addiction, OCD, eating disorders, and depression. So how can we study these apparently important systems? Most of our knowledge come from animal models where we can do invasive studies and where we have techniques with high spatiotemporal resolution. But what about humans? Do we want to study these systems in humans? And I think and hope to persuade you that the answer is yes. Human brains are different. The basic neuromodulatory machinery may be conserved across species, but it operates on different neural systems in the human brain. And human cognition is different, and I think this point is particularly important when we want to speak to complex clinical conditions that are hard to reproduce in animal models. But just like animal models can be used to validate human work can be used to validate animal models. On one hand, human researchers like myself often get criticized for not really studying neurobiology as we know it in animals. But on the other hand, animal researchers get criticized for not really studying cognition as we know it in humans. So there seems to be a lot of merit in being able to ask the same questions in both humans and other species. So my point that human brains and human cognition are different does not mean that the roles of dopamine and serotonin necessarily will be different. It just means that we should really go test it. So how can we study these neuromodulators in humans? As I alluded to earlier, we don't have the same kind of methods as in animals, and a common theme is low spatiotemporal resolution. For example, a drug may take hours, days, or even weeks to have its effects. There are a handful of pioneering studies that have directly recorded from dopamine neurons, but these recordings can only be part of the story. A single dopamine neuron, shown here, projects to hundreds of thousands of targets, and at each of these target sites, you might have local modulation of dopamine release. But, and this is what I'll talk about today, we can now use fast-scan cyclic voltammetry in the conscious human brain. And it's quite a long word, so I'll just call it voltammetry for the rest of the talk. Why is this exciting? Well, voltammetry allows us to monitor electroactive brain molecules such as dopamine and serotonin at very fast timescales. And this means that we can begin to understand how the information encoded by neuromodulatory systems are transformed at the downstream sites of release during active thought and behavior. We've had the opportunity to do these measurements in patient volunteers who are undergoing DBS surgery for management of Parkinson's disease or essential tremor symptoms. Now, the areas that we can record from are naturally constrained by the surgical procedure. Just like when you're hitchhiking, you can only ask to be dropped off along the plant route. And thus far, we've made recordings in the caudate and in the putamen, depending on the surgical trajectory. And we've focused on the striatum because it receives dense projections from dopaminergic neurons in the midbrain, and because the striatum supports the fundamental cognitive processes that are believed to be shaped by the dopamine system. Now, instead of taking the scenic route and going straight to the results, I'll spend some time explaining what voltammetry is and how we have adapted it for the human brain. Why? Well, separating dopamine and serotonin in voltammetry data is thought to be a real challenge. So I want to explain why we are able to do this and why you should have confidence in our results. So how does voltammetry work? You take an electrode, typically a carbon fiber electrode, and you insert it into a wet lab or brain environment where you'd like to learn more about different neurochemicals of interest. You then raise and lower the electrode potential multiple times each second. And each of these voltage sweeps induces electrochemical reactions that depend on the identity and the concentration of the brain molecules that are present at the electrode tip. And this is the good news for us. We can measure these electrochemical reactions as changes in current. So in this way, we can use the current responses to monitor electroactive brain molecules at very fast timescales. We've had to take several steps to adapt the technique from rodents to humans. And one of them has been building carbon fiber electrodes for the human brain, primarily increasing their size. And in this first paper, Ken stimulated dopamine axons into the rat striatum and showed that the current response measured on our human electrodes is similar to that you get on a standard rodent electrode. But perhaps the biggest change that we have made is our approach to signal inference. And just to be clear, I don't mean the actual data acquisition, where we use protocols similar to those used in rodents. I mean how we go about making inferences about dopamine and serotonin after we have made the recordings in the patient brains. Now, one inherent constraint in voltammetry is that we have to use data collected in a wet lab environment to make predictions about data collected in the living brain. So we want to create a statistical model that uses the in vitro data in the best possible way to make inferences about the in vivo data. The advantage of animal models is that we can do controlled experiments to corroborate the output of the statistical model. We can use the same electrode for in vivo and in vitro recordings, and we can stimulate the release of specific neurochemicals into the recording site. But we can't do these experiments in humans. So what we did using insights from machine learning was to develop a statistical approach that makes robust out-of-training predictions and thereby partially alleviates the need for validation experiments. Now, to achieve this, we train our statistical model using data that are several orders of magnitude larger than those typically used in animal studies. And more specifically, we collect training data on multiple electrodes, and for each of these electrodes, we cover the range of biologically realistic concentrations in small increments, and we make multiple measurements for each concentration. And just for those of you not familiar with chemical abbreviations, DA is dopamine and 5-HT is serotonin. And here are the exact numbers. So overall, the training data is pooled across 20 electrodes, and each of these electrodes contributes 5,500 samples. And this gives us a total of more than 100,000 samples. And just to put this into perspective, a traditional voltammetry study may only use a handful, 5 to 10 samples for each neurochemical of interest. And what we do next is to use the labeled training data to fit a multivariate regression model that makes concurrent predictions about the different neurochemicals of interest. And another difference to the classic approach, for those of you familiar with voltammetry, is that we use the raw data rather than its principal components for all of these steps. So here we have an example measurement. On the left, we have the triangular voltage function applied to the electrode every 100 milliseconds. Here in the middle, we have the current measured during the application of the voltage function. And on the right, we have the differentiated current response, which is what we use for signal inference. And the reason why we use the differentiated current response is that we have multiple electrodes in our data, and using the differentiated current response removes potential offset terms due to subtle variation in those electrodes. So why do we use the raw data instead of its principal components? Well, we do this because we've seen that information about dopamine and serotonin is distributed throughout the entire current response and not just at singular locations. Here, the different colors correspond to different dopamine and serotonin concentrations, with dopamine on the left and serotonin on the right. And as you can see, the different concentrations separate and even invert across the voltage suite. And crucially, this step is supported when we look at the dopamine and serotonin parameter vectors that we get from our statistical model. So here I've taken the regression coefficients for each time point and using a blue-red color scale, plotted them on top of an example current response from one of our patients. And what these plots tell you is that our statistical approach found that almost every single time point can be used to separate and estimate dopamine and serotonin. Okay, so now we have our statistical model and we're ready to make predictions about data collected in the brain. But before we do this, let's look at how the model performs when we apply it to withheld data which we collected in the wet lab, but which we didn't use to train the model. And the idea is that this step mimics the out-of-probe scenario that we face when turning to the patient recordings. So we've done lots of these model evaluations, and you should check out our paper for more information. Here I'm showing data for 3 electrodes D, E, and F, where we varied both dopamine, shown in blue, and serotonin, shown in red, over time. And the solid lines indicate the true concentration and the dots indicate the model prediction. And as you can see, our statistical model does a good job at separating and estimating the 2 neuromodulators at the same time. Okay, so this was perhaps the more boring stuff. So now that we have a measure, a way to measure dopamine and serotonin, what do we want to know? Well, the starting point for our first set of papers was the role of the striatum in reward-based learning and decision-making, and in particular the role of dopamine in value coding. A central hypothesis in neuroscience, first formulated by Reid Montague and Peter Dayan in the 90s when they were postdocs with Terry Sarnowski, is that dopamine signals a reward prediction error, the difference between the reward that you expected and the reward that you actually got. This error signal is extremely useful because it can be used to learn how valuable different features of the environment are and which actions are more valuable to take in the long run. Moreover, which is interesting to us because we can measure serotonin, people have proposed, like Peter Dayan, that serotonin carries the opposite signal to dopamine, coding reward prediction errors in a negative rather than a positive frame of reference. So to test these hypotheses, Ken Kishida and Rosalind Moran asked patients in the surgical suite to play a sequential investment task in which they had to bet on stock price fluctuations in different historical markets. Now, you may ask, why didn't you start with a classical conditioning task or a 2-armed bandit or some other task we know from the animal literature? And the answer is we did, but it was too boring and too tiring for patients, so we switched to a more engaging task. Now, because the markets are not fully predictable, you're going to experience outcomes that are better or worse than expected. In other words, positive and negative reward prediction errors. And Ken and Rosalind found that on a large proportion of trials, dopamine and serotonin tracked these outcomes in the predicted manner. Dopamine increased for positive reward prediction errors, whereas serotonin increased for negative reward prediction errors. Okay, so we have found evidence in support of roles for dopamine and serotonin in value coding. But this task that I just showed you is a low-dimensional probe of the dopamine and serotonin systems. It only involves a single valence axis ranging from punishment losses to reward gains. And recent work suggests that these systems play a much broader role. For example, we have evidence from human and animal studies that both systems are involved in action, with dopamine invigorating action and serotonin inhibiting action, extending their hypothesized opponency to the action domain. But even in the value domain, dopamine and serotonin are not always opponent. Serotonergic neurons can be activated by reward predictive cues and by both positive and negative reward prediction errors. There's also evidence that dopamine and serotonin track other dimensions than value and action, such as uncertainty about the outside world or learning about sensory statistics. So what we did next was to study dopamine and serotonin in the context of perceptual decision-making, where variables relating to sensory inference, choice formation, and behavioral control are all at play. And the idea is that by using more complex or more multidimensional tasks, we can better understand the contribution of dopamine and serotonin to thought and behavior. Now, an issue with many standard perceptual decision-making tasks is that they don't fully tease apart these different component computations. So take the classic random dot motion task that many of you might know. So here you have to say whether a field of dots is moving left or right. And this decision is made easier or harder by changing the proportion of dots that move coherently relative to the proportion of dots that move randomly, noise dots if you like. But it can be difficult to interpret what a neural response means in this task. So let's say we find that when coherence goes up, dopamine goes up. But does dopamine track uncertainty about the sensory stimulus or the difficulty of the decision, which can be influenced by other factors? Or does dopamine trigger the behavioral response, which can be prepared more quickly when coherence is high? So to overcome these limitations, we used a modified version of the random dot motion task that I developed while I was a postdoc with Steve Fleming at UCL a couple of years ago. So here's what we did. First, we used a continuous range of motion directions and not just left or right. So here the angle of dot motion relative to the center is about 5 degrees, but it could have been 45 degrees or any other direction around the full circle. Second, we asked subjects to judge the motion direction relative to a variable reference direction, which only appeared after the motion stimulus had terminated. So in this example, the correct response is to press the orange button. The motion direction is clockwise relative to the reference direction, but you cannot know this beforehand. So the idea is that in this way we break the relationship between sensory inference and choice formation, because you cannot prepare a decision until you've seen the reference direction. In addition, we manipulated experimentally the demands on sensory inference and choice formation using a 2x2 factorial design. First, we varied the fraction of coherently moving dots in order to create trials characterized by different levels of sensory uncertainty. And the idea here is that the lower the coherence, the higher the sensory uncertainty. Second, we varied the distance between the motion direction and the reference direction in order to create trials characterized by different levels of decision difficulty. And the idea here is that the lower the distance, the harder the decision. Okay, so what this factorial design means is that there will be trials where you felt fairly certain about the motion direction, but where the decision turned out to be hard. And trials where you felt fairly uncertain about the motion direction, but where the decision turned out to be easy. So what does behavior look like on this task? Do our experimental manipulations work? So to answer this question, we're going to look at choice accuracy. And we'll use the x-axis to indicate distance, low and high. And we'll use separate lines to indicate coherence, with low coherence in blue and high coherence in red. So here we have the data from the 5 patients in our study, 2 Parkinson's disease and 3 essential tremor patients. And indeed, coherence and distance have independent effects on their performance. Choice accuracy is higher A when coherence is high. The red line is above the blue line, and B when distance is high. Both lines have a positive slope. And reassuringly, if we average across all trials, the patient's performance falls within the range of healthy controls here indicated by the gray bands. And finally, if we look at data from a pre-surgical visit a couple of days before the surgery, we see that patients' performance is well matched before and during surgery, really demonstrating that we can run these kind of tasks in the surgical suite, a rather special setting. Okay, so now we know that our task works behaviorally. So we're going to look at our measurements of dopamine and serotonin. And I'm going to structure the results according to where we recorded from in the striatum. In primates, but not other species, the dorsal striatum is divided into the caudate and the putamen by a white matter structure known as the internal capsule. And in the rodent literature, this roughly maps onto dorsomedial striatum and dorsolateral striatum. We have evidence to suggest that these 2 structures are part of distinct functional loops. a cognition loop passing largely through the caudate, receiving inputs from association areas, and ultimately returning. Sorry, I just have to close this. A cognition loop passing through the caudate where you have inputs coming from association areas and where outputs are ultimately returned to prefrontal cortex. And on the other hand, an action loop passing largely through the putamen, receiving inputs from sensory motor areas and ultimately returning outputs to premotor areas. We recorded from the striatum in 4 patients and the putamen in 1 patient, and we're going to leverage this feature of our data to unpack the contribution of dopamine and serotonin to active thought and behavior. Okay, we're first going to look at the caudate data, and we're going to ask whether dopamine and serotonin track the cognitive variables probed by our factorial design. And we're leaving out one of the 4 caudate patients for reasons that will become apparent shortly. Now, the dopamine and serotonin responses may reflect a number of task variables. So take this example trial. Does the serotonin response reflect coherence, distance, the interaction, choice accuracy, or choice reaction time? We don't know. But because we have dissociated these task variables by design, we're going to be able to answer this question. And we're going to do it using a regression-based approach. So more specifically, we're going to do a sliding window regression. So here we have our design matrix or predictors, including our task variables for each trial. We fit this design matrix to the dopamine and serotonin responses sliding through time, running the regression model at each time point. And this procedure returns encoding profiles, beta time series that quantify the relationship between our task variables and the neuromodulatory responses. So here, the vertical lines indicate the onset of the motion stimulus and the onset of the reference direction. And the colored lines indicate the encoding profiles, or beta time series. Pink is coherence, blue is distance, and green is the interaction. And overall, we don't see much happening in the dopamine response. By contrast, for serotonin, there's a strong negative effect of coherence during the presentation of the motion stimulus before the reference direction is shown. So, what this analysis shows is that serotonin in the caudate tracks our manipulation of sensory uncertainty. And we see this pattern not only at the group level, but also in individual patients, including PD and ET patients. So take the second patient, the Parkinson's disease patient. When coherence is low and sensory uncertainty is high, serotonin increases. And when coherence is high and sensory uncertainty is low, serotonin decreases. Now, what about this mysterious 4th caudate patient? Here, both dopamine and serotonin appear to carry information about the task variables at the start of a trial. And this really puzzled us. But after having ruled out timing errors during data acquisition, we began to wonder whether dopamine and serotonin were perhaps sensitive to experience dependencies between trials. In other words, did the systems pick up on any coincidental structure? So our task turns the classic random dot motion task into a discrete 2x2 design, crossing coherence and distance. This gives us 4 trial types. And each of these may be followed by another of the same 4 trial types. And the question is whether the dopamine and serotonin systems picked up on coincidental structure in these transitions. The trial types were randomized, but the experience probability of transitioning between 2 trial types may seem non-random. And this is something that happens to us all the time. If I flip a fair coin 5 times and it's heads every time, you might wrongly think that you can predict the next outcome. So here's an example of the experience probability of each trial type on the current trial, given that the trial type on the previous trial was defined by high coherence and low distance. And in this example, one of the trial type transitions is less likely than the others, and it would therefore be more surprising from a statistical learning perspective. Interestingly, it turns out that both dopamine and serotonin track the experience task statistics. So here the x-axis shows the transition probability of the current trial from least likely to most likely, and the y-axis shows the dopamine or the serotonin response on the current trial from the smallest to the largest. And in case you're wondering, these relationships hold after controlling for the marginal probability of each trial type, and as well as sequential effects on choice accuracy or choice performance. And I'll get back to what these results might mean at the end. This is not something we set out to test with our task, but we found it interesting, so we reported it in our paper. So finally, we're going to turn to the patient where we measured dopamine and serotonin in the putamen. Unlike the Caudate patients, we didn't see any encoding of the task variables, and this fits with the proposal that the Caudate Putamen division reflects a cognition-action axis. But the question is whether we see any action-related effects in the Putamen data. Are the dopamine and serotonin responses aligned to the moment at which the patient implements their choice using the gamepad? And the answer is yes. Here we have the dopamine and serotonin responses averaged across all trials, and the pink histogram indicates the distribution over choice reaction time from the slowest to the fastest choices. And as you can see, dopamine and serotonin track this distribution, with dopamine ramping up and serotonin ramping down. And in line with the notion of a cognition-action axis, we didn't see this response pattern in the Caudate recordings. Okay, these results suggest that changes in dopamine and serotonin delivery to the putamen may trigger the execution of a choice. And if this is what happens, then we would expect these changes to happen earlier for faster choices. So to test this hypothesis, we're going to take our trials and, using terciles over choice reaction time, categorize them into fast, medium, and slow trials. And the prediction is that the changes in dopamine and serotonin happen the earliest on the fastest set of trials, here shown in pink. And this is what we see. So here we have the average dopamine and serotonin responses for our 3 choice reaction time bins. And as you can see, dopamine peaks earlier on faster trials, shown in pink, whereas serotonin drops earlier on faster trials. And that these responses are tied to action is even more apparent when we align to the moment at which a choice is made, the moment at which a button is pressed. And here we see that regardless of the time taken to make a choice, dopamine ramps up, whereas serotonin ramps down immediately before a choice is made. So to use a car metaphor, it looks like dopamine plays the role of the accelerator, triggering movement, whereas serotonin plays the role of the handbrake, holding movement and then allowing it to be released. Okay, so these are the results I was going to show you. So what I'm going to do now is take stock and look at the bigger picture and talk about some future steps. So we found evidence that serotonin tracks uncertainty about a short-lived stimulus. And I don't think this has been seen before in any species. A number of animal studies have linked serotonin to uncertainty on another class of tasks, reward-based decision-making, where animals have to learn from experience which decision, in this case left or right, is most likely to produce a reward. For example, serotonin neurons track not only signed reward prediction errors, whether an outcome was better or worse than expected, but also unsigned reward prediction errors. And these tell you how surprising an outcome is, irrespective of whether it's better or worse than expected. So the fact that we see this computational motif, if you like, across domains and across timescales suggests that serotonin may encode a generalized uncertainty signal. Why is this a useful signal for the system? At fast timescales, uncertainty is an important signal for behavioral control. So if you're driving through the rain and you can't really see what's in front of you, it's probably a good idea to slow down. And at slightly slower timescales, uncertainty is an important signal for learning. So take this scenario. The left option has led to reward in the past, but now you take the left option and you get nothing, a big reward prediction error. Now, this tells you that you can no longer be certain about the task rules, the world might have changed, and perhaps you should change your model of the world accordingly in order to make better choices in the future. And we do have causal evidence that serotonin drives plasticity. For example, if you stimulate serotonin neurons on particular trials, it turns out that those trials have a bigger influence on an animal's future behavior. And really broadening out, I also think this is one of the cases where we can speak to clinical implications. SSRI, selective serotonin reuptake inhibitors, which increase serotonin levels in the brain, are a common and sometimes very effective treatment for mental health conditions such as depression and anxiety. And perhaps the reason why SSRIs can help break rigid thoughts and behavior is not because of any direct effects on mood, but because of general uncertainty-induced plasticity and learning. A surprising aspect of our data was that dopamine, at least at the group level, didn't really track our task variables. Why is this surprising? Well, we know that on the classic random dot motion task, the firing rate of dopaminergic neurons track motion coherence. So why didn't we see this? Well, it might have to do with the disease state of our patients. It might have to do with our factorial design, which breaks the direct link between motion coherence and choice accuracy. Or perhaps it's because we didn't provide trial-by-trial feedback, a reward signal if you like. But if you look into the supplementary materials of our paper, you see that the dopamine levels do fluctuate across time. We just can't make sense of them using our factorial design. So this is more speculative, but perhaps the reason is that dopamine reflects more specialized computations. For example, coding for particular parts of the stimulus display or particular stimulus-action pairings. And we do have reason to believe that this might be the case. And in this recent paper, Ben Engelhardt, who happens to be our host today, showed that distinct clusters of dopamine neurons encoded distinct aspects of cognition and behavior. So in our case, perhaps we shouldn't expect to see anything clear-cut when we average across physically very different instantiations of a particular task condition, and especially not at the group level where we also average across different sites of dopamine release. Another surprising aspect of our data, which we hadn't set out to test, was that dopamine and serotonin tracked experienced task statistics in one of our patients. Now, our task doesn't allow us to isolate the underlying mechanism. The responses could reflect some cognitive construct that is modulated by the task statistics, such as dreading or hoping for particular trial times, given what you've experienced this far. But they could indicate a more direct role for dopamine and serotonin in statistical learning about the outside world. This statistical learning interpretation fits with work from Jeff Schoenbaum's lab, indicating a role for dopamine in learning about sensory statistics. So these are statistics that are not directly tied to value. So this is my example of their rodent task. So let's say I prefer Fanta and Coke by an equal amount. In other words, Fanta and Coke have the same value. What Jeff Schoenbaum's lab has shown is that dopamine neurons track not only changes in value, as we expect, for example, getting more Coke than I expected, but also changes in identity, for example, getting Fanta when I was expecting Coke. In other words, a change in the sensory features of a reward, but not a change in its value. So this result is interesting, but there's also a potential issue in the sense that value and identity are yoked. For example, we can't say for certain that Fanta and Coke are of equal value in high-dimensional space. I might prefer one over the other at particular moments in time. So we're therefore planning to run a task where we fully separate the sensory statistics from reward. And the basic idea is to have a task where the start state fully predicts whether you're going to get a reward, but where the probability of different sensory experiences along the way depends on the start state. So let's say I get into my car to drive to Starbucks. I could do this during the day or in the evening. In both cases, I'm going to get my coffee, my reward. But the sensory statistics of the trip, of the car journey, the probability of what I see out of the window will depend on the time of day. And the question is whether the dopamine and serotonin systems track these expectations and signal violations of what we expect sensorially, irrespective of the reward we're going to receive at the end of the journey. finally, we found evidence that both dopamine and serotonin responses in the putamen were closely aligned to the moment at which a choice was made, suggesting that they're directly involved in triggering action. Both results fit with recent animal work. For example, optogenetic stimulation of rat dopamine neurons increases the frequency and vigor of self-paced movements. Conversely, optogenetic stimulation of rat serotonin neurons increases the amount of time that rats, or perhaps it was mice in this case, are willing to wait for an uncertain reward. So in summary, I hope to have convinced you that dopamine and serotonin are not just involved in value coding. Instead, they support a diverse range of computations. And with that, I thank you for your attention.
All right. Thank you very much, Dan.
Well, thank you.
Note, we're having a technical issue with the group chat. So please, for asking your questions, please chat Mariana Duna directly. You can find her by going to your chat tab on the left-hand side, then searching for her name, Mariana, and then chatting her directly your questions so she'll be able to read them. These issues are not being fixed, so it will be fixed for the next talk. But for this talk, you can just chat your questions directly.
Yeah.
And while we're doing that, I actually have a question for you, Dan, which is there's a lot of different issues that you've discussed that potentially could influence, for example, the dopamine response. One of them is the action, as you talk about the action trigger. There's also the issue of values, and also the issue of test statistics, and of course the timing. There's this nice paper from Joe Patton Lab talking about how dopamine also relates to timing. And so obviously it's a difficult issue, but in general, how can we disambiguate all these different roles? When you can't do everything in a single task. Right? So how are you thinking about this multiple roles of dopamine and how to disambiguate them?
Well, I think you're right that you can't study all of these questions in a single task, but I do think that the way forward is using more high-dimensional tasks like you have done yourself, which we've tried to do here using a dynamic perceptual decision-making task. Because that's perhaps going to better allow you to parse out the different aspects of the dopamine response that you might otherwise confuse in a low-dimensional task where the value of an option, the time it takes you to go and collect that option, the value statistics are all sort of bungled together, if you like.
Okay, we have a question from Genevieve actually, and it's how do you determine if a decision is easy or hard? And thanks for the interesting talk.
Okay, yeah, thank you. So we determine whether a decision is easy or hard using our task. So I'm just going to load up the behavioral figure. So what you see here is that choice accuracy, patient's performance is higher when distance is high and when coherence is high. So we use these two task factors to assign difficulty to a decision, which is a function of both of these factors. Now you could say this is just choice accuracy. Are people aware of this? And the answer is yes. And I didn't present this today, but we also, because we're studying humans, we can ask for this. We also ask them to estimate confidence in their decisions. And we see that people's confidence shows the same pattern of results. So people's confidence reflects the impact of our task conditions on choice accuracy.
Okay, thank you.
I don't know if that answered the question.
Yes, it did then. Thank you.
All right. I think Mariana is anymore. So just as a question, why did you choose to give reward to the patient during the task? It seems that maybe you could have added the reward skill that you could expect to your list of variables to this degree?
So your question is why we didn't give people rewards in the task?
Yeah, I think to remember that you didn't. Is that correct?
Yeah, that is correct. So, I mean, now, well, in our original task, we didn't give people rewards because we wanted to study sort of the subjective sense of uncertainty, subjective sense of confidence. So the idea was that we could look at confidence responses without those responses having been trained up on some reward schedule. So we used that again here. And I think the idea was that we were interested in what do these systems do when we don't have a reward schedule, incentive structure painted on top of the task. But now in hindsight, it would also be interesting to run the task with reward, as you say, because it might be interesting to study how reward might back propagate through the dopamine response to earlier parts of the trial, the extent to which they are predictive of the upcoming reward. But the idea as a first step was to use a task where reward wasn't painted on top of it, to sort of go beyond that value coding aspect of it. I mean, it might still be there. It might still be subjectively rewarding to feel like you're getting something right.
Right.
Okay, so I think we are out of questions so far.
All right. Maybe I can ask another question.
Yeah,
Go ahead.
So, I mean, this is really, this technique seems amazing to me because obviously we've been using, rodents and monkeys because that's where we can use this technique. So now you have this working in humans, which is really opens the door to a lot of things. And one of the things that I'm interested in is the possible spatial specificity of the dopamine response. So do you have a sense of how much you're integrating when you're doing voltammetry in humans? How much of a big of an area are you integrating the dopamine concentration in the dopamine?
Well, I mean, so it's sort of extracellular. So you're sitting sort of outside the synapse. So you're obviously picking up you're potentially picking up dopamine release from multiple downstream neurons. I can't remember the exact specifics. So probably looking at I might get this wrong, but maybe you're looking at like a micron of a centimeter. Is that what you had in mind? Sort of like.
Yeah. So just in a sense of like what percentage of the, let's say, the putamen you're measuring the dopamine release, just because it's not clear in humans. We have no information, right? But it might be because we have such big brains that you can actually have sort of different dopamine concentration in different parts of the structure.
I see. Yeah, so you're measuring it. You're not really looking across the entire putamen.
Right.
So you're looking at a very small area inside the putamen. So I guess something I didn't speak to here is where are we in the putamen or in the caudate? Because if you study the rodent striatum, it does matter, right? Whether posterior, anterior, or how dorsal, how ventral you are. So now I think we've run studies on about 100 patients. So I think that because we have the coordinates for each of those patients, we can start asking questions about location within a particular neural structure. But you're looking at a small area, but obviously you're not looking at dopamine at one particular synapse. If that makes sense.
It does. I mean, that's actually, it would be amazing to see if you can map out, the location versus the response and try to see whether you have some sort of like a sort of topography or some sort of spatial structure of response for the different variables in different locations in humans. That's an amazing data. I think everybody would be very interested to look at.
And I think I should also mention, so this far we've done recordings in conjunction with DBS surgery. But we also have proof of principle work suggesting that that you can read this information of other electrodes than a carbon fiber electrode. You can read it off, for example, the platinum iridium wires that go down into the brain when you do epilepsy monitoring, for example. So there you have patients who come in, they might be in for a week as recordings are made at multiple time points. So that also opens up for the kind of tasks you can do. You can have a bit more movement. You can perhaps have something more immersive than you can in a surgical theater. So that also means you can begin to look at other structures that are interesting but don't lie along that pre-planned surgical trajectory down into the STN or where the neurosurgeon is going for the DBS surgery.
Yeah, that all sounds like very, very exciting directions for field research. So I'm very much looking forward to see what you do with that.
Yeah.
It really opens the door to a lot of things that we're only able to do with maybe rodents to this point.
Yeah, exactly.
So just a note, so the chat is working now. It has been fixed. So now if anybody has a question or a comment, you can use the chat. Please use the stream free chat so we can all see your you can all see your questions and your comments. And if you have a question for Dan, please write it now. We'll wait 1 more minute and then we'll introduce, if you have more questions, we'll introduce our next speaker.
I mean, so now if there's that time, then I'll also just fill it in a little bit. So another thing we are looking to do is not just to look at dopamine and serotonin, but really see how far we can push this machine learning approach. Like, so what if we use other neurochemicals such as glutamate or acetylcholine, and in particular noradrenaline, and we have proof of principle work to suggest that we can look at noradrenaline as well at the same time as dopamine and serotonin. So that's something I'm working on now is to have that model trained up on training data, including noradrenaline, and then go back and reinterrogate the data I've shown you now. And it might be that's what looks like the dopamine signal, or the dopamine signal doing nothing, is some kind of combination of dopamine and noradrenaline, because they have similar current responses. So it could be that when we add noradrenaline into our model, that we can then start seeing something we couldn't see with the data I just showed you now.
That's amazing, because the classic idea is that you can't separate neurology from dopamine.
Yeah, because I think it's because the classic idea is very much based on looking at the current responses. So if you look at the current responses, do they look different for the different neurochemicals at different concentrations? And what's definitely true is that I cannot sit and look at the current responses and see what I'm looking at right now. So perhaps it does make sense to use a machine learning approach where you really just see how far you can go in terms of predicting what information is in there. So I think a lot of the views about what you can and cannot separate come from the more traditional electrochemical approach, where you do your measurements and then look at those current responses and then make decisions as to whether you can separate or not.
All right. That sounds, again, it sounds incredibly exciting that you can do all these things in humans. And I'm very much looking forward to see what else you do with that. I mean, it really opens up a lot of really, high-level cognitive tasks where we're actually looking at very precise measures, what seems to be very precise measures of the different chemicals. So thank you so much for the great talk.
Yeah, thank you very much. So I'm going to